and welcome to the Absolute Recap Biology Edition, where we aim to maximize your understanding and minimize your need for memorization. Each episode will recap content, skills, and test-taking tips to help you succeed in May. I'm your host, Melanie Kingett, and your recap starts now. Hi, and welcome to the Absolute Recap Biology Edition. Today's episode will recap mitosis. Let's zoom out. We're in Unit 4, Cell Communication and Cell Cycle, Topic 4.6. Our big idea is information storage and transmission. What did the cell say when her sister stepped on her foot? Mitosis? Corny, I know, but appropriate pronoun association. Whereas we use masculine terms with genetics problems, paternal, F1 generation for filius or son, it's all about the ladies with cell division. We will have daughter cells and sister chromatids. Cells don't have gender, but this terminology may stem from early cell studies in which larger-sized egg cells were used. Let's zoom in. Mitosis, the process of nuclear division, which creates two identical diploid cells. Think the T of mitosis for two. Remember that diploid cells, 2N, are somatic body cells with chromosomes in homologous pairs, one inherited from mom and one from dad.
Magnificent!
Sandwiched between interphase and cytokinesis in the cell cycle, the entire process takes approximately 2 hours with a series of coordinated chromosome movements. Mitosis can also be referred to as karyokinesis, or division of the karyotype. A karyotype is a mapping of chromosomes, organized into homologous pairs. Karyo comes from the Greek, meaning kernel or nut, and has the same association to the nuclear material as the term eukaryote. So, mitosis, dividing the nut. Mitosis has four main sequential events: prophase, metaphase, anaphase, and telophase. These can be recalled simply with the mnemonic PMAT. These events arrange and divide the chromosomes evenly, if all goes well, towards opposite poles of the cell. Each of these phases has discernible features when viewed under a microscope, but the cell doesn't pause when it reaches each step for a snapshot. This is why you will often see terms like pro-metaphase or meta-anaphase showing transitional images in diagrams. Before we recap each step, let's remind ourselves what the cell has accomplished thus far. Interphase is complete, with cellular growth and organelle duplication achieved in G1 and G2. DNA has also been copied during S phase, and the cell has passed all checkpoints. It's safe to assume that the cell is ready to begin nuclear division.
Hello, AP.
So we'll just leave this blank for now, and I'll come back and say the subject name here.
Student, I know that you are currently preparing for your AP
The subject name.
exam, but are you taking more than one AP course? Maybe you want to get a jump on courses you plan on taking in the future? Do you think that you're better than me? Is that what you think? Because I'll have you note that along with the Absolute Recap
The subject name.
edition, we have podcasts and study guides for biology, chemistry, music theory, physics 1, and U.S. government. So if you think you're better than me, if you are currently taking or plan on taking more than one AP course, visit www.TheApsoluteRecap.com. That's www.TheAPsoluteRecap.com. And click on the courses link or search The Absolute Recap on any podcasting platform. And now, back to the Absolute Recap
The subject name.
edition.
P. First is prophase, in which the nuclear membrane and nucleolus break down. DNA becomes more visible as chromosomes coil and condense from loose chromatin into their familiar X shape. The extra copy of DNA that was formed in the S phase can now be identified. Each half of the X is an identical sister chromatid connected to each other at the centromere. Additionally, spindle fibers form from centrioles at centrosomes, if you're an animal cell. Plant cells will have an alternate version of microtubule organization for division. PM. Next is metaphase, where the goal is to line up chromosomes in the middle of the cell, known as the metaphase plate. Not an actual plate, just an identified cellular location. During metaphase, some spindle fibers extend from the centrioles and attach to kinetochores at each centromere. The centrioles have migrated migrated to opposite poles of the cell, and through a tug-of-war motion, align the chromosomes to the middle or equator of the cell. This is a big checkpoint for the cell, and division will not continue unless all spindle fibers are attached. Other spindle fibers extend to the opposite pole, giving structure to the division process. PMA. During anaphase, sister chromatids are pulled apart, separated at their centromere. As spindle fibers shorten, the identical copies migrate to opposite poles of the cell. Other spindle fibers extend and elongate the cell's shape, and since they are being pulled from the middle of a chromosome, each end tends to bend, and so you can even see little capital A's in diagrams if you look from the right angle. PMAT. Finally, telophase. We need to undo the steps of prophase. So we will uncoil DNA back into its loose chromatin form, reduce spindle fibers and centrioles, and reform what is now 2 nuclear membranes and nucleoli. Oftentimes, cytokinesis begins while anaphase and telophase occur. Remember, cleavage furrow for animal cells and cell plate for plant cells. Okay, great. So we have 2 new cells. What was all that for?
What's this all about?
Growth, tissue repair, and asexual reproduction. Each new daughter cell has a complete genome from a parent cell. Time for unit connections. Big compare and contrast with Unit 5 and meiosis. Also, strong connection to Unit 6, gene expression and regulation, watching out for errors and their effects on phenotype. All right, what about the exam? Be prepared to explain how mitosis results in the transmission of chromosomes from one generation to the next. You may be expected to predict the effect on a cell if there is a disruption in the cell cycle, or checkpoints. A common exam error is forgetting to explain the purpose and timing of the cell cycle checkpoints. You should also be prepared to answer a comparative question about mitosis and meiosis. I highly recommend using diagrams that illustrate chromosome movement. Better yet, sketch and caption your own. To recap, mitosis is the process of nuclear division that creates 2 identical daughter cells. Through PMAT and the splitting of sister chromatids, more cells are created for growth, repair, and asexual reproduction. Coming up next on the Absolute Recap Biology Edition, meiosis. Today's question of the day is about cytokinesis. What protein filament creates the contractile ring in animal cell cytokinesis? For the answer to the question of the day, please follow us on Instagram at TheApsoluteRecap. That's the APsolute Recap. Check out our website, TheApsoluteRecap.com, for episode schedules, study guides, virtual tutoring, and to sign up for our virtual classroom. The Absolute Recap is produced by Brad Kingett with music by Zach Caruso. Today's episode was written by me, Melanie Kingett. Thanks for subscribing, and don't forget to rate and review wherever you get podcasts. Time's up. Pencils down. Thank you for listening to the Absolute Recap Biology Edition. AP is a registered trademark of the College Board. Copyright 2020, Absolute Recap LLC, all rights reserved.